Good evening, guys. Is my voice audible? Can you guys
Yes,
hear
sir.
me now?
Yes,
Yeah,
sir.
yeah, yeah. Good evening to all, and happy to see you all once again after a weekend. And I welcome you all for this Advanced Bioinformatics course, Day 6. Is my screen visible?
Yes, sir.
Yes,
So,
sir.
yes. So for people who have joined recently to this course, welcome you all for this course. So I guess you're aware of the syllabus. So far we have seen about introduction to bioinformatics. So in that we discussed about overview of advanced bioinformatic techniques and their application, a role of softwares and other tools in bioinformatic research, and emerging trends and challenges in the field. And secondly, we started with this next topic called next generation sequencing data analysis. And on that, we have completed pre-processing and quality control of NGS data. And we have seen a demo of FastQC, a tool that we use to quality check the raw data, which is provided by the NGS vendors, that is NGS data analysis, sorry, NGS sequencing. So today we'll see about alignment and mapping of the sequence reads and variant calling and structural variant analysis. We'll see what are the other tools we are going to use, and I'm planning to design a workflow or a demo series if possible. We'll just make it happen in the next class. Okay. And we'll start with the class now. So you guys clear?
Yes, sir.
Yes, sir.
So please let me know whether my screen is visible now. I've started presenting the screen.
Yes, sir.
Yeah, that's great. So before jumping into the session, how many of you have tried this fast QC? Try installing the Ubuntu WSL and trying installing your thing, this fast QC and running it. So many of you have tried to install. So it's great. People have tried to contact me and they encountered some problem, but it is great that you guys tried. So I appreciate you guys. Yeah, Rajat, you raised your hand. Saurabh, yeah.
Yes, sir. I
So
was just
you,
telling I installed it.
yep. So you guys tried installing and what are the problems you encountered, or were you guys successful in running the process? The recording started already. It is recording. The session is recording. Yeah, thank you, Firoz. Showing to you guys whether it's recording or not. Is it showing?
Yes, sir.
Yeah. Okay.
Yes, sir. It's recording.
So okay. Yeah, please let me. Let's have a small interaction now on this fast QC. Some of you guys have tried. So please tell me your experience or tell me the problem what you encountered. Anyone?
Sir, actually, I tried to install fast QC. Then
Okay.
some problem comes. Then I
Okay.
search on ChatGPT how to install. Means I give the problem. They give provide me the solution. Means how to code, how to means everything I got from ChatGPT, and I have successfully installed fast QC,
That's
sir.
good. That's good. Yeah, that's nice.
Sir,
So,
sir, I actually downloaded the Ubuntu and the fast QC file through Ubuntu, but sir, I was unable to download the SRA file that was through
okay.
the NCBI.
You're trying to download SRA file instead of FASTQ file?
Sir, first it was recommended to download the SRA file, and then I saw on YouTube and he said that then convert the SRA file to fast QC
Okay,
file.
you follow the video tutorial which I sent you, right? which I
Yes,
showed
sir.
you.
Yes. Yes.
Okay, that is also one of the way where if you're try downloading SRA file format instead of FASTQ, there is an intermediate step where yeah, we have to take in account. So that step is called SRA toolkit. So that's a tool.
Yes, sir.
SRA
Yes.
toolkit is a
SRI
tool,
toolkit.
and yeah, so we have to rely on that tool in order to download a SRA and converting it into a fast queue. But the thing which I showed you in class, it is that in that we can directly download the fast queue file instead of SRA file.
Sir, if we
Uh,
want to download any file from PubMed or NCBI, so we can directly download to FastQC, or first we have to convert through SRA toolkit.
yes, that is possible. So that is what I showed you on that day, just downloading fast queue file instead of SRA file. It is a direct approach. Okay, that day I showed you like two options where to download FASTA and FastQ, right? So that is the direct approach. So you could have done that, and you can try copy pasting that file format into the home directory of your WSL. That is the Windows, that is Linux environment.
Yes, sir. Yes,
Okay,
sir.
so that can be done. Okay, I'll just show
Okay.
you later in the end of the class how that can be done. Okay, so
Okay, sir.
and you're aware of the commands, right? This fast QC commands. So it is good that when you're working in terminal, if you don't know the steps, one thing you can just rely on some open forums like Quora and some open forums like GitHub. In GitHub repository, we have discussions or something called Discord. We have. Apart from that, in those forums, your question may be answered soon, or it may not be answered soon. And in that case, you can rely on ChatGPT, and mostly the data which is provided by ChatGPT is accurate. So you can rely on that, and we are here to help you. So by that you can do. Okay. Yes, and you guys face any other problem? Any other problem? No? Okay, so not an issue. So in terminal, just pretty much you can get help, or you can get to know what that particular tool does with with a command called hyphen hyphen help or hyphen hyphen h, which shows you what that particular tool does. For example, when you're running fast QC and you don't know the arguments what to pass, in that case, what you can do is you can just type fastqc space hyphen hyphen help. So this particular command works on all the terminal, pretty much in all the terminal softwares. Okay. So not only bioinformatic tools, in other open source tools and other generic tools also. So they will be having a stipulated section to help you out. So it will be showing what are the options can be enabled when you're running a tool through terminal. Okay. So let's quickly jump in. Lastly, we have seen about this pre-processing and quality control. Next thing is we're going to see about alignment and mapping of sequence reads. So before that, I just wanted to add on a point. So since this is a quality control analysis, FastQC is a quality control analysis, you have to run it multiple times in order to verify the quality of the sequence or quality of the data what you have. Okay, so it is not only the first step; it should be the intermediate step and the last step. So, for example, when you're converting the SRA file into a FastQ file, you will be running and you'll be taking the primary data, the data which I showed on the HTML file, that will be there, and you'll be interpreting the results from there, and you'll be trying to remove the adapter sequence using Trimmomatic and some other tools called FastP like that. And after trimming and after removing the adapter sequence, you can also run the same fast QC, that is quality check on the same file. So you can check whether the particular HTML file, the output file is changed. So that is what quality check is it? So when you're doing an analysis, you can have a thorough quality check on the file on each and every step. So you can do multiple quality checks, like soon after converting the file, and soon after trimming the file, and soon after aligning or mapping the sequences, and soon after doing all this thing, you can have a final report. So these reports will be helping you in refining the data, refining the FASTQ file or the NGS data. So this is the second step, which is so crucial: alignment and mapping of the sequence. So you'll be having a raw data and you'll be checking its quality, and with that quality data, that quality results, you'll be knowing what to do next. So if the sequence has any adapters, if the sequence has any SNPs, that is single nucleotide polymorphisms, or if it has any other variants, both normal variants and structural variants, what you could do is you can just remove it. You can make that particular data more presentable and more accurate. Is okay. So this is the second step. So this most important step in analysis of NGS data, that is alignment and mapping. So this comes to in picture when you have a raw data. You know what the organism, from where the organism you have taken from, but that organism is not well defined, or when that particular organisms is possibly could be a subspecies or substrain of anything, right? So we don't know the taxonomy and the hierarchy. So to verify that, mainly to verify that, and other things also there, mainly to verify that the particular sequence can be brought up from this particular, say, genus or species, we could be doing this. Okay, we should be knowing what are the chromosomes, from which position of the chromosome this genome is taken. In this case, if it is a whole genome, so we should know its origin, right? So we should be knowing its pakka origin, its clear origin. So to identify
Yes,
that,
it
yes,
actually. Yeah,
sorry,
your screen is up blank from my phone.
your phone. Okay.
Yes, sir, actually.
Can you see my screen whether it's changing or not?
It's not even changing,
Yeah, we
sir,
can see.
during.
Okay, I guess you're having some network issue from your side. Uh, uh, can you do one thing? Just leave the class and kindly rejoin so that your network gets restarted or reset,
Okay, sir. Okay, sir.
or else yeah, try doing that. Okay. Others could see my slides changing, right?
Yes,
Yes,
sir.
sir.
Yeah. Okay. Okay. And yeah, please try doing that. Okay. Yeah. So let her join. So yeah, Basha, is my screen visible now? Is it changing? Is the slide changing?
Yes, sir.
Yeah.
Yes,
Okay.
sir.
Yeah. Thank you. Thank you. Thank you. So where did I leave? Yep. So this is one of the crucial step to identify the origin of the sequence, from where the organism or the gene of an organism is established, and how well the sequence is performed, and what is the phylogeny of the thing, or phylogeny of the organism. And in multiple aspects, we are just going to align and map the sequences. So how it is done? You'll be having this mapped genome already. It is called a reference genome. When you run a whole genome from an organism, it will be published in databases. It will be stored in databases so that we could cross-refer with the whole genome. So that we'll be having a clear-cut picture of what we're working with. Okay, so we should be having a reference genome to where we'll be aligning our sequence, our data. So when it comes to wet lab, also you'll be when, for example, if you're doing antimicrobial assay or antibacterial assay in a distribution method, you'll be having a control in order to analyze your compound, in order to compare your compound or your nanoparticle along with the control, right? So in that case, we'll be having a clear-cut idea on how much the zone of inhibition happens. So likewise, we'll be having a reference genome or reference sequence data, so that we'll align our data to it, and we'll check whether it is the close related species or close related species, and whether it is having any relation between the previously updated data. So the importance is essential for reliable variant detection and downstream analysis. Downstream analysis where you process the data, you produce the data in a clear cut manner. It should be in a proper manner, and this aligns sequencing reads to reference genome to determine their origin in the sense, their what organism it is, what is the genetic markup of it, and what is the chromosome number till that, okay, and much more. This process involves mapping the sequence trees to a reference genome or assembly to determine the original genomic location. Genomic location is where you karyotype the chromosome, and inside chromosomes will be having a long sequence of genes which is highly bound to a histone protein, and yeah, genetics pretty much. So to know its original genomic location, and what we should know this close relation between the species, we'll be doing this alignment mapping, okay. So common alignment algorithms are BWA, Burrow-Wheeler's aligner. Last time we have seen about this. So this particular algorithm works in a idea of full text minute index. So index is what the same index what you have in a book. So indexing helps you to find the particular sequence of a data or particular region or particular base of a NGS data. So that is what an index does in a book, right? So if you wanted to refer a book, a specific detail in a book, you will just go to the index and find it out, right? So that is what this aligner or this algorithm works. Is cross refers with the resources which is already there, pre-available data, and it finds the quality and it aligns according to the previously available data, because the previously available data are the well-defined data, and it is processed already, and it is there already, like it will be in up to the mark level, okay, that quality level. So pretty much all these softwares follow that FM index, so it will be fast and accurate in aligning. So we discussed this pros and cons earlier, high accuracy and speed, but the repetitive regions will not be identified. It will be having high repetitive regions, like SNPs will be more or will not be will or it will not be identified. And bow tie is also one of the same kind of tool, but we don't have that high repetitive region, that kind of struggle in this bow tie. Okay, it uses a full text minute index for a fast and a memory efficient alignment. So it aligns and maps the sequence. That is, uh, for example, I'll make it simple for you guys. You'll be running blast, right? So there'll be a subject and a query where you'll give the subject and it queries along with what are the data which is there in the thing. So it is just a pairwise alignment. Likewise, this is also your NGS data will also be aligned in order to the reference genome. There you will be aligned or it will be cross verified with all the data which is there in the repository, which is there in the database, NCBI database, and it'll be giving you the first 100 or first 200 kind of thing. But here you will be having one or two reference genomes where you will be aligning your sequence to it, okay. And where you can use it, it'll be helpful in aligning short reads to a large genome. So when you are having a short kind of NGS data, short reads of data, and you can use bowtie to compare and contrast it with the large genome data, that is the reference data. Okay, and next thing is called star. If it comes into picture when a special type of sequence data is there with you. It is called the RNA-seq data, RNA sequence data. So initially we seen about this types of sequencing, right? NGS data sequencing. So one of the thing is RNA-seq. So it can be used for RNA-seq data with ability to detect the splice junctions. And the advantages is high speed and highly sensitive for RNA-seq alignments. Okay, you can rely on the tool called STAR for RNA-seq alignments. Okay, so what are the parameters influencing alignment and quality? So does someone have any questions?
Sir, what is the full form of star? I mean, is it having a specific full form?
Yes, it does have. Just a minute. So can you guys hear me now?
Yes, sir.
Yes, I'll just
Yes,
put
sir.
it in the chat box. I
Yes, sir.
muted myself and started talking. Okay, this star is also called as splice transcripts alignment to a reference. So as I told before, in RNA sequence alignment data, uh, this splicing will be happening. Like it is the most crucial step in post-transcription modification, where we'll be removing the introns, non-coding sequences, and we'll be binding the exons together, the coding sequences together, which is called splicing. So this mutation is normally we make that. This the pre-mRNA complex, or which has both introns and exons, will be spliced, and this particular mRNA will be sent for a sequence. So on that case, we can what we can do is we can just use a star, the spliced transcript alignment of a reference tool in order to cross verify with the pre-available RNA-seq data. Okay, whether the particular genomic data or the particular RNA data is fully consists of exomes, the coding region, or it has any SNPs or any other additional base pairs to it. Okay, so yeah, Varshini, have any question?
Yes, sir. In the last class, you talked about the fast QC, right, sir? And
Yes,
this alignment, is this the step after the
yes,
NGS that happens?
it is after the quality control.
Okay, sir.
So that day we discussed about the steps involved in uh, the analysis, right? The whole NGS data analysis. The first step is fast QC, running fast QC and analyzing its quality. So quality
Okay,
checking,
sir.
quality control. Next thing is alignment and mapping.
Okay,
Okay.
okay, sir. Got it.
So once you know the quality of the data, what a particular vendor or what you got from a machine is, then only you can jump into other steps, right? So that is what I mentioned. We can run fast QC, not only the first step, it can be the middle or it can be at the end to even and check the efficacy of the data what you have after trimming, after aligning it with a cross-reference genome, and kind of finally coming to a conclusion that the file what you have is the best. Okay.
Okay, sir.
So I'll just break it down. First, you'll be having a fast Q data. Okay. First, when you're trying to do NGS data analysis, they'll provide you a raw data. It is in FASTQ file. So what we'll be doing is we'll just have that raw data. Since it's a product of sequencing, uh, there are high chance or high possibilities for that sequence to have nonsense base pairs. Nonsense base pair in the sense it is not like it is useless, but apart from the ATGCs, the base pairs, deoxyribonucleotides, it will be having other nucleotide properties like other alphabets R, T, Q, S, N, M, like that. So before running FastQC, what you'll be doing is you'll be referring IUPAC ambiguity codes in order to change the particular non-ATGCs to convert it into a ATGC, right? Those kind of nucleotides. This is the pretty much first step you are going to do. But the thing is, nowadays, like recent advanced technologies will be helping you where the data given to you, the raw data given to you will be itself well refined. So the person who's giving you the data will convert the IUPAC ambiguity codes according to the IUPAC norms, and he'll give you the data. Okay. So earlier, we used to get data like along with this nonsense nucleotides. Okay, so we have to do it manually. But nowadays, people who are doing this sequencing, no, they'll be given the data after converting that ambiguity codes. So pretty much our first step to do is to check the quality of the raw data which is given. So we'll check the quality check. We'll run the quality check. We'll have the first report, and after that, if we are finding that we are having this SNPs, single nucleotide polymorphs, that is addition or deletion of any other nucleotides within the region, or there is the adapter sequence which is not removed from the raw data, or any other possible errors, will be doing that, will be rectifying and will be modifying the data, right? So that is what Trimmomatic does. We'll be trimming the adapter sequences and nonsense sequences from it, and we'll make it refine. Okay, so that the reduction happens there in the sequence data. And next thing is, you'll once again run the fast QC, so that the previous data. Yes, I'm talking. Ekta, you couldn't hear me. I guess people are having this problem, network issue. Okay, so yeah, just a minute, guys. Someone couldn't hear my voice. I'll just rectify what's the problem.
Sir, actually, you are audible. I think the problem is at
Problem is
there.
with that person.
Yes,
Yeah.
sir.
Yes, yes.
Okay. So yeah, we'll continue the session. So you'll be running multiple fast QC. So in order to, when after trimming the sequences and removing the adapter sequence, you'll run a fast QC. And after doing that, when you get to know uh, your particular data should be aligned, or if your particular data has any other nonsense sequences, or your data which has this match and mismatches, in that case, you have to do the particular approach for it. And after doing the alignment also, you'll be running the fast QC file, and you'll be running this fast QC throughout to make the data more refined. Okay, so that is what fast QC. It is not only the first step. It is going to be there on it, it is the alternate steps of all steps. Okay, so once you done fast QC, you'll be doing this adapter trimming, and you should run fast QC, and you should do alignment. And one more time, you can run this fast QC, or you can run multi QC. Multi QC is the tool where you align or you quality check multiple data at the same time, multiple fast files at the same time. We can do that too. And once the alignment is also good, what we should do is we'll be saving that particular file, the fastq file, into a file called sam file. So it is sequence alignment map file. So it'll be having the extension sam. So that will be done by the tool called sam tool. Okay, clear till now?
Yes, sir.
Yep, that's great. Yeah. So parameters influencing alignment quality is mismatch penalty. The penalty of mismatches between the read and the reference we can be identified, and the gap penalties, penalties of insertion or deletion. So when a nucleotide, particular nucleotide is inserted or deleted, sometimes the particular section or the particular position will be marked with hyphens, or there won't be any nucleotide present over there, so it will be as a gap. So we can identify that. And based on the alignment scoring, so we'll be having this alignment scoring. So it'll be that weighing the alignment based on the match, mismatch, and gap penalties, we'll be having a score. So these are the parameters you commonly see in the alignment tool. It is the same parameter where you see in a blast, or where you see in megax, that is multiple sequence alignment also. So you guys have run this blast, right? Blast tool. You're familiar with blast tool? So
Yes, sir.
you've heard this kind of
Yes,
jargons there: mismatch penalty, gap, alignment scores, e scores, e values, like that. So you'll be taking those same parameters here in this alignment tool also. Okay. So next things we have to make sure when you're doing this alignment is this accurate read placement. That is, alignment ensures that each sequence read is correctly placed on the reference genome. This is fundamental for downstream analysis such as variant calling, expression quantification, and identification of structural variation. So the thing is, the reference genome what you have should also be up to date. It should be more refined. It should be the data, because that is the data which you're going to use as a reference. So it should be in proper alignment, and the thing, the NGS data what you got as a result is should be also refined properly, right? So once the alignment is proper, it is so fundamental that once the alignment is proper alone, we can just go on to the next steps like variant calling and expression quantification, like how well that particular gene got expressed in this series, like that, and identification of structural variations, and so on. So this is the first thing we have to keep in mind. Next thing is quality assessment and filtering. This aligned data provides metric that helps assess the quality of the sequence experiments. Okay, so it we can tell this is the metric unit. Okay, where you'll be having particular scores when the alignment is done, and it's called alignment scores, so that you would be knowing that how well the sequence is aligned, how well the sequence is refined also. Okay, so things we have to take in account is mapping quality indicates the confidence that a read is correctly aligned to the reference, and coverage in depth shows the distribution and numbers of reads mapped. That is, mapped in the sense it is aligned properly with the same, with the consecutive or complementary basis, like that is called mapped. Okay, to the region of the genome, which is essential for understanding the completeness and uniformity of the sequence. Okay, so our job is to refine the data and to make the data what we have, which is more resembled to the reference genome. That is what it says. It is so simple. Okay, so identification of genomic variants. So why alignment is important in identification of genomic variants? Like, alignment allows to detect the single nucleotide polymorphs. This there is nothing like you'll be having this insertion of single nucleotides like A, T, or GC, which is repetitive in the sequence. We can't identify when the sequence is enormous, right? So alignment helps us to identify in which place newly this nucleotide got added or inserted. Okay, insertion, deletion, or indels, and large structural variation can be identified, and these variants are crucial for studying on genetic diversity. So when it comes to genetic diversity, I told you, like when you have a whole genome sequence of an organism and you have a short genome sequence, which you have done doing this NGS next generation sequencing, you'll be taking that whole genome sequence and comparing it with your data, right? So in that case, there are high possibility ways or steps to identify the genetic diversity, the phylogeny of the organism, whether this is the particular strain or it has substrains, or this particular thing has the same diversity, like that, and disease association, and also evolutionary biology. Okay, so next thing, to analyze gene expression, we could do this alignment, or else this alignment will be helping us in gene expression. Okay, for RNA-seq data, the alignment is so necessary to quantify gene and this transcripts. So DNA to RNA is transcripts, right? So this transcript abundance, accurate alignment of the transcriptome or genome enables the identification of differentially expressed genes. So we can't say that the genome which is present in a chromosome or in this particular loci will be expressed the same way it expresses in the different organism of the same type. Okay, so we can't tell that. That is what evolutionary biology is. That is what genetics is. Okay, so due to some mutations, it can be modified. Due to some parameters or some insertion or deletion of a single SNPs, it may be modified, or some modifications in the coding sequences, that is exons, may be resulting in differential expression of genes. Okay, a gene which is expressed in me may differentially expressed, or it can be expressed or may not be expressed in another organism, right? And like my fellow human mates, like that, and isoforms revealing insights into a gene regulation and function. Okay, so alignment is so crucial, where you can use or you can take this help of this alignment or mapping data I can go all the way along to the gene expression analysis. So it is the primary key feature of this alignment and mapping. Okay, and error correction bias reduction. Aligning the sequences helps in identifying and correcting sequences error and biases. So I told you when we run it in a machine or we'll be having this human errors maybe, or the buffer which we use, or the washing buffer, or the chemicals which we use, all this may sometime interfere with the result, and the result may be biased. Okay, so this misalignment can highlight systematic errors, the sequencing or the library preparation. I guess you guys are aware of the UMIs which we talked about, unique identification markers. So it will be helpful in the library preparation. So we talked about this library preparation bias, right? So this alignment will be helpful whether we had any bias at that time where we prepare this library, right? So at that time we could possibly identify where we had this kind of problem, if the problem is there. But most probably we don't encounter this kind of problem usually, because this is more well refined formula, and the formulations are well refined, and the steps are well defined, and the technological advancements, and the high rec 2500, the Illumina's new machine has lot of noise elimination and error rectification softwares running behind. Okay, so there are no possible chances, but it is good that you're also checking this error correction bias reduction also. Okay, so tools used for alignment and mapping, we discussed this already. So BWA, that is Burrow-Wheeler aligner. Detailed workflow is indexing the reference genome, aligning reads, and generating SAM or BAM file. Okay, SAM file is what I told, the sequence alignment map. Okay, and BAM is binary alignment map file. Okay, the binary alignment map is like where there is less information of the alignment mapping file. SAM file, it will be more detailed. Likewise, where you have FASTA and FASTQ, right? Same like that, SAM and BAM are pretty much same file formats of the FASTQ or the alignment maps. But the thing is, BAM is binary, so that it has less information, less characters, and SAM is the more detailed one, more elaborated one. Okay, so the output will be SAM and BAM files containing aligned reads. So these alignments will be of directly aligned to the reference genome to the query. Okay, and bowtie 2 is the tool also used for indexing, alignment, and post processing of aligned reads. Post processing of aligned reads here is refining and removing the introns or any other mutation which happened in the data what you have. So special features are suppose end to end and local element modes. Okay, local element modes is where you snip and you have a small reference genome which can be used in order to organize and align your small data. Okay, when you have a small sequence data, you can index a small reference genome to it. Okay, and next thing is star. So spliced aware alignment indexing and multipass alignment can be done here. So it is highly sensitive and accurate when it comes to RNA sequence data, and for detecting splice junctions where whether the particular sequence has only transcripts of coding regions, not introns like that. And visualization after doing this, after generating this alignment map file, whether it's SAM or BAM file, what you'll be doing is you'll just load that file into integrative genome viewer. It is the online engine, or we can call it as a online workflow to visualize the aligned reads in a genomic context. Okay, you can provide or you can produce graphical data using this integrated genomic viewer. Okay, so next thing is variant calling and structural variant analysis. So soon after doing this alignment, we'll be doing this variant calling and structural variant analysis. So far, is it clear? You guys any have any other doubts?
No, sir.
Okay. Yeah, Vashni, you raised your hand.
No, sir, I just forgot to like raise.
Okay, okay. Yeah, okay. That's great. So you guys able to follow me? Yep. Okay. Toshani, sorry if I misspell your name. So it's just a disclaimer, and if you guys just don't feel bad when I mispronounce your name. Yeah, tell me, you've raised your hand.
Yes, sir. Sir, I was asking, can you please repeat when IGV is used?
IGV, Integrated
Yes,
Genome Viewer. So you're asking for this IGV when we are using this IGV tool. So Integrative Genomics Viewer is a online platform where you can analyze genomic structures and genomic sequences. You can do multiple things on the genomic side, but here what we're going to use this is when you align and map a reference genome along with your FASTQ file, and you'll be having the alignment file in the form of SAM or BAM. Okay, sequence alignment map file or binary alignment map file. So there are other tools where you can visualize the data and you can make it more make it more presentable in a graphical stature. And one of the tool is IGV, where you can visualize the aligned data, aligned sequence reads, and you can go through it. You can do multiple analysis, post analysis after aligning it in a genomic context. Okay, so it comes under after aligning and mapping the sequence with your reference genome. Okay, so it is for you visualization of your data, your BAM file or SAM file, aligned data. So pretty much when you're aligning the sequence itself, you'll come to know whether the data what you have, the sequence data what you have uh, is correct, is up to the quality, is up to the rank. So you can eradicate the positions. You can check the positions, and you can eradicate the single nucleotides within or at the end or something like that, and you can snip it off. And you can likewise snipping the adapter sequence in the trimmomatic, that is the previous step. Like that, what you can do is in this alignment and mapping, you can snip the SNPs and you can make it as a SAM file. Okay, so you'll know it is like a railroad. Both are complementary, like the subject and the query are complementary, so that the gaps, the mismatches, and the SNPs, the nucleotides which has got inserted, or the nucleotides which got deleted, or any other possible errors can be rectified when it is properly aligned. So that is the idea. Okay, you guys clear?
yes, sir. Thank you.
Yes. So next thing is variant calling and structural variant analysis. So this is where the SNP comes into picture. So definition for variants are SNP is a variant. Okay, just keep that in mind. SNP, single nucleotide polymorphisms, single base pair changes in the genome. Okay, it is not a high res jargon. Don't need to worry about that. SNPs are some nucleotide which got inserted in a coding region of a genome. Okay, it may be on any position that called single nucleotide polymorphism. Usually, for example, there is a sequence called ATTGCA. It is the original sequence, and here, when SNP got added, it will be ATTGCCA. So you'll be having couple of C's there, like that. Some kind of single nucleotide got added to the genome, where it makes a possible error, or it will be resulting in the data which is not in a good quality. Okay. So we don't know what is the read made up of or what is the sequence made up of. We'll be identifying it as the good sequence, and we'll be moving forward with the data, right? So that shouldn't be happening. The correction, we'll be having gigabytes of data, where you'll be having lakhs and lakhs of nucleotides. Okay. In that, we won't be knowing whether this particular nucleotide or nucleotide base pair is right one, right? Okay, so this step is helping us to identify that kind of errors. Okay, so indels or insertion and deletions, where a nucleotide or set of nucleotides, a couple of nucleotides got inserted or deleted, can be identified. It is also called variants. And structural variants or SVs, in that case it is a large scale genomic alteration, such as duplication or deletion or insertion or translocation. You have studied about these structural variants in your biotechnology course, maybe. You guys heard of it? Duplication, deletion, insertion, and translocation.
Yes, sir.
Yes, sir. Yes, sir.
Yep.
Yes,
It
sir.
was there in
So
with.
Yeah, so
Okay.
those are theory part, and this is where it comes into practical. Okay, so these are called structural variants. So it'll be influencing your data, your particular sequence in high manner. It will be influencing your data where you won't be having the proper data then like that. Okay, so some duplication may be happening on same loci the different sequences or in different loci like that, or deletion may be happening, a segment of gene or segment of sequence got deleted, or nonsense kind of sequence may be inverted or made different or translocated. Okay, so these are the possibilities in genetics where we should take in account. So ultimately we are playing with genes, we are playing with genomes. So these are the parameters or possible things we should note it down. Okay. So steps in variant calling is read realignment. So you have a alignment mapping. So you'll be having the same SAM file or BAM file, and you'll be realigning it and checking for indels, insertion, deleted insertion and deletion nucleotides, and also for SNPs. Okay, so around indels to improve accuracy using tools like genome analysis toolkit. And inside genome analysis toolkit, we have lot and lot of tools, and one of the tool is called realigner, indel realigner. Okay, so previously alignment and mapping was done, and you'll be having SAM and BAM file in your hand. Before that, to analyze the fast QC, you'll be having fast Q file, and you'll be converting it to SAM file or BAM file after doing the alignment and mapping using this tools what we talked about, star and this aligner tools, BWA and Bowtie 2. And when it comes to variant calling, what you'll be doing is you'll be checking for this indels, insertion and deletion kind of segments, or any other SV structural variants, the large kind of large segment of DNA sequences. So this can be done on GATK or Genome Annotation Toolkit. And inside Genome Annotation Toolkit, you have various of tools to do that. And one of the tool is called Indel Realigner. And the name itself suggests that it identifies any insertion and deletion and realigns the SAM file or the BAM file what you have. And next thing is a base quality score recalibration. This one we talked about earlier, where you also have a GATK, a tool, PQSR tool to adjust the base quality scores. Okay, once the indels got eliminated, or once the SNPs got eliminated, or once the large nucleotide sections, the structural variants got eliminated or rectified, the quality score will be improving. Okay, so we should be recalibrating, and you'll be having the best quality scores in order to take this data to the next level. Okay, so when you run a blast, you will be selecting its data, or so you'll be selecting the FASTA file according to the e value, right, and the accuracy and the percentage of similarity. Likewise, we also have a base quality score where it should be best where after doing this process and all. Okay, next thing is variant detection, identifying variants like SNPs and other deleted variants, are the variants which you saw in the previous slide, like any possible actions like duplication, deletion, inversion, and translocation, which happened in the particular thing. We can't say, or we can't just confirm whether these variants happened in the alignment, that is the previous step, alignment and mapping step. We can just identify whether the sequence is extra. We can assume that yeah, some SNPs are there, or we can say some structural variants are there. But in order to analyze and in order to remove it, or in order to do something with the data, you should be doing that in genome annotation toolkit software, and haplotype caller, or FreeBayer's or SAM tool itself. And FreeBayer, SAM tool, both of these tools are terminal based tools, and haplotype caller and genome annotation toolkit are GUI based, graphical user interface based. Okay, so structural variant analysis involves identification of the characterization of structural variations in the genome. Okay, likewise, this deletion, duplication, and insertion, translocation can be identified. Okay, the structural variations are large-scale alterations which is happening in the DNA that involve segments of genome, typically more than larger than 50 base pairs will be deleted or translocated or inserted like that. So these variations can have significant impact on the gene functions and regulation. Okay, we can't say that the gene or the sequence as a gene what we have is the appropriate gene which does a job. Okay, for example, we'll talk about this eye color. The gene which is responsible for eye color is there in my sequence, but I couldn't tell that the particular sequence is refined. Okay, so maybe this particular well, let's say that the gene which is responsible for eye color is more than 300 base pairs. Okay, so in that, this 50 base pair or larger than 50 base pair structural variance, if it is present, I couldn't tell that whether it is the original genome or original data what I have, or whether there's there any structural variance present over it. Okay, so to analyze that, so whether when there's a structural variant present in the gene, it will be having a high impact whether it will be upregulating the gene or downregulating the gene, causing it it'll be having the expression or the differential expression of the genes, right? So these variations can be highly significant impact on the impact of gene functions regulations, contributing the phenotypic diversity, not the genotypic. Phenotypic is where you can visualize, we can see clearly with our own eyes what happens, the changes. Okay, and in evolution status, okay, and also in disease susceptibility. So types of structural variants, there's a list. Here is the list. So deletion, when 50 or more than 50 base pairs got deleted, loss of DNA segment, or duplication, additional copies of DNA segment. Maybe the same 50 base pairs or more than 50 base pairs will be duplicated in the same sequence or in the same sequence at the different positions. Okay, and inversion, a segment of DNA is inversed end to end, like for example 5 dash to 3 dash will become 3 dash to 5 dash, like totally inverted. And translocated rearrangement, where a segment of DNA is moved from one location to another, either within the same chromosome or the different. Okay, so this can be happening. So let's mark as position 1, 50 base pair or more than 50 base pair or some set of gene should be there in the place, will be relocated or translocated to the different position. Okay, that is called translocation. And insertion, that is, you all know about it, addition of extra DNA sequence into the genome. Or copy number variation is difference in the number of copies for a particular DNA sequence. Okay, so these are the variants, structural variants you should take in account when you are doing this NGS data analysis. Okay, so variant calling, structural analysis, structural variant calling analysis, what are the tools we can use? This is first one is MANTA or MANTA, and detection of structural variants, including large insertion, deletion, and complex events, may be identified and rectified. And lumpy is the integrated structural variant analyzer, and it is a framework which we can use for multiple signals from aligned reads. So when you have a alignment map, that is SAM alignment data, you can put it into this lumpy, and you can analyze the structural variants. Okay, and Delhi is the tool which we can, or the algorithm which we can use it for identification of structural variants, including translocation inversion. So we have different structural variants, types of structural variants, and we have different tools for that too. Okay, so it'll be easy for you guys to remember. Okay, so tools for variant calling and structural analysis. Genome Analysis Toolkit is the web-based engine where you will be having, it's the comprehensive toolkit for variant discovery and genotyping. And within that, you'll be having Halotype Caller, Mutex2, base recalibrator, and BQSR, like that, and Intel recalibrator, Intel realigner, those kind of tools. This genome analysis toolkit, well, the name insists that, like the name tells that it is a toolkit where it has collection of tools to it. Okay, so following GATK for genome kind of works is the best practice for variant discovery and ensure accuracy and reproducibility. Okay, and free bias. So it's the Bayesian genetic variant detector for calling SNPs, indels, and complex variants. So when you have done using this GATK for deletion, duplication, inversion, and translocation, you also use this free bias for identifying SNPs, indels, insertion, deletions, and complex variants, other variants, and usage scenario suitable for population level variation analysis. Population level is where population genetics comes into place. You have large amount of NGS data, population data with different races and with different organisms. So in that case, you can use freebase to call SNPs. This is where you load up multiple files at the same time and you can cross verify. Okay, and next is SAM tool. SAM tools is that day I showed you in my terminal. I also use SAM tool. It is the suite of program for interacting with the high throughput sequencing data and identifying and edit, or you can manipulate the aligned sequence which you got from the BWR bowtie tool. Okay, and Manta, it is the structural variant caller. It takes large scale of SVs, that is structural variants with high sensitivity, and integration is works seamlessly with other variants calling and alignment tool. Okay, you can integrate this Manta to bowtie or sequence alignment mapper tools in order to call. It's kind of a collaborative tool. Okay, and Lumpy, we discussed about it. Identification of structural variants, use split trees and discordant read pair signals, and it is kind of a pipeline where you design a workflow and Lumpy will be part of the pipeline. Okay, and is that the software as we discussed so far? BWA is the alignment of sequencing needs for a reference genome. SAM tool, those, so it is just repetitive. Okay, we can just go through it. And recap is primary data analysis. What you do for quality checking? It's the high quality reads. You'll be identifying it, quality control. And if there is any low quality bases or adapter sequences, you'll be doing that trimming. And once that is done, you'll be doing mapping and alignment so that to identify whether it has any variance or any structural variance and whether the data is apt to the reference genome. And next thing is variant detection. If the data or the genomic data what we have has any variations or variants called SNPs or any other insertion and deletion indels and other structural variants, we'll be identifying that. So peak calling is identifying regions of the genome where the read are enriched, such as ChIP-seq experiments where you have this. NGS data can be done in within the chips. Okay, and data visualization is what we talked about is IGV visualization of the results and analyzing in order to gain insight into the data and identifying the potential biological significance of this aligned sequences what we have or post aligned sequences, that is the uh, GATK tools. Okay, and further analysis can be also done on this GATK and other associated tools also there, like performing additional analysis steps such as gene expression analysis. Gene expression analysis is where the particular set of genes in your genome, whether it is overexpressed or highly expressed or differentially expressed, or it is expressing in the proper way, like that, to gain a deeper understanding of the biological process involved. Okay, so I've shared you the document already, right? Have you guys went through this links? Gone through these links? Yes. Am I audible? Or is my mic on mute? Guys?
Yes, sir. Yes, sir.
Yeah.
Yes, sir.
Okay. So please go through the links. Okay, I've provided you the resources. Just go through. Since those are websites, they'll be having a about page. Just read and understand it. If you guys have any trouble in understanding the particular tools, or if you have any trouble in understanding the methods, please let me know. We'll just discuss it later. Okay. So I've given all these links. Please do visit these links. Okay, or if you guys needed any tutorials based on this, I'm happy to help. Okay, so so we have done with the second chapter or second topic, NGS data analysis. Okay, I just don't want to dump you with all the analysis what we do on NGS data, but this is the most prominent and most important set of analysis what we commonly do with the NGS data. That is the file what we have. Okay, apart from that, we have this post data analysis and post sequencing data analysis, which will be overwhelming for you guys. Okay, so if you guys wanted, I'll share the resources and I'll just tell you about it later. Okay, yeah, Ramya, you have raised your hand.
Sir, what is the difference between SAM file and BAM file? And is there something that one file is more efficient than other file?
So you're asking about the map files, right?
Yes, sir. SAM and the BAM files that have taught earlier.
Okay, so SAM file is sequence alignment map. Okay, and BAM file is binary alignment map. Okay, and sequence alignment map is where the reference genome and your query, the data what you have is properly aligned with lot of data. That is a lot of data in the sense, lot of information. Okay, whether the thing is properly aligned, whether the sequence is having the mismatch alignment, or it has gaps, or it has the best quality scores like that. Okay, and BAM file is just binary. It just aligns the data in different positions or in the single position, like query start and end positions. Okay, it just gives a transcript or a kind of a binary file, like a pairwise alignment file. Okay, but SAM file is more advanced. You'll be having informations like mismatch alignment, gap junction penalties, and quality scores will be marked in the SAM file.
Yes, sir. Thank you.
Yeah. Anyone else have any questions? So, and regarding one of you guys have asked whether you guys will be having a question like a test or exam like that. So the answer to that is you'll be having a MCQ kind of test at the end of the course. Okay. In order to qualify this course, you'll be having a MCQ kind of a test. Okay. You will be conducted, and once done, you'll be qualified and you'll be certified with this. Okay. And most probably all this Saturday and Sundays will be off. And we'll extensively discuss about those stuffs on the weekdays. Yep. So how many of you guys have just had a look to the previous links which I shared to you on the previous slide decks? Like we talked about this Galaxy, Taverna, Apache softwares like that. We discussed about those, right? Have you guys had a look?
Yes, sir.
Okay, so that is great. So try to understand what it says, okay? And I'm here to break it down and make it understandable, okay? And if you guys have any other doubts, we'll just discuss it now. Okay, it's already 9:10. Yeah, Vashni, tell me.
So, could we have some like practical sessions, sir, like where you are, running an analysis or something, and we also like do it in parallel with you
Yes,
in
I'm
the
thinking
meeting?
about that. Yeah, I'm thinking about that. Since this is an advanced bioinformatics course, we encountered a problem earlier, right? When I thought of showing a demo, running everything at the same time, this Microsoft Teams and the thing the same time. We like it is not that prominent, but I am planning to show you some demo and I'm planning to give you some hands-on so that you'll be having a better understanding. Okay, so what I was instructed is to just teach you guys, and with this 1 hour time frame, I could just think on that alone. But yeah, we could have a practical session because this is something what we should do practically. I do believe that. So that is why I asked you guys to run this fast queue on your weekend. Okay, so uh, uh, please try that. Please try this fast queue running fast QC file. I mean fast QC analysis. That'll be great when if most of you guys achieve that. We can just go on to the next steps. That is trimming the non low quality sequences and adapter sequences. I'll teach you that too.
Sir, I
Okay.
did try, but I wasn't able to install Fast
Since
QC.
this is your first time, you will be having a lot of problems. So, well, installing Ubuntu is not much of a deal where if you
Yes,
have
sir.
a Microsoft shop, Microsoft store, from that you can install because that is the
Yes, sir.
Windows subsystem for Linux, WSL. So that won't be a problem. So soon after installing, it will be prompting for you to give an username and a password. So
Yes,
this
sir.
password should be so simple. So all Linux passwords will be commonly as root, ROOT. You can set the same password because it is a huge pain when you forgot the password what you set and uh, try remembering it, and it is so hard to reset the password too in Linux platforms. Okay, that too it is so hard in WSL. So keep the password simple. This is the first
Yes,
step.
sir.
Next thing is there are different methods in downloading the data. Okay, something we usually do is called wget. Wget is the method where you have a link, and if you wanted to download a particular thing from a link, we will use this wget space and we'll copy paste the link. So in our case, it is the FASTQ file. So we'll just locate where the FASTQ file is there in the web, and we'll copy the link. And in Linux, what you'll do is you'll just create a folder. Folder, how you'll create? You'll just type mkdir space, and you can rename the folder as test or analysis like that. So mkdir is the command to create a folder. Okay. So inside, I guess I have to teach you this common commands, right? You guys not aware of the common commands?
Yes, sir.
Okay, so let me try showing my terminal. I don't want to take much of your time. I'll just show what are the common things. So sorry. So is my screen visible now? Just tell me whether when my screen is visible.
Yes, sir.
Okay, so this is the WSL, the Ubuntu terminal which is running on in my Windows. So first thing is I'm the I'm the administrator Sam at the rate Sammy, and it will be commonly in the uh, commonly it will be there in the mm, home folder, home directory. So you don't know whether which directory are present. What you have to do is you can just click on PWD, that is present working directory, and click on enter. It says, yeah, you're at home. On that, there is a directory called SAM. Here, there in that particular directory. Okay, and just wanted to visualize, or I just wanted to see what is there in the SAM folder or directory. I'll click on LS. LS is list, and click on it. It will be showing what are the folders and what are the files what I have here. Okay, so someone is waiting outside. So next thing, ls and mkdir. So to create a folder, mkdir and it'll be called as test. So I already have a folder called test. Let me create analyze ana analysis. Okay, and I press enter. It creates it. So how do I check that? I'll type ls, and you can see it is there. Can you see this folder? It got created. Okay, so what if when I wanted to delete something? So what I have to do is rm dir, that is remove directory, and I'll type analysis, enter. So when next time when I go and check that, it'll be getting deleted. Okay, it don't be there. Okay, so from this SAM folder, I wanted to go into different folder. So what should I do then? I'll be typing cd space. Let's say I have to go to the test directory, test, and enter. It goes to the test directory. How I come to know that? You can see this test is here. Okay, and I have to view what is there in the test folder. I'll once again click ls and enter. It shows what is the thing here. So these are the common uh, uh, commands what we use in Linux platform. So to clear the workspace, to clear uh, the section, what I'll do is I'll just type clear, and if I press enter, it clears the window for me. Okay, and if I wanted to go to the previous folder, what I have to do is cd space double dot and enter. It goes to the previous folder. So these are the common commands we'll be using. So I just wanted to invoke, or I just wanted to start a tool. We'll start fast QC. What we'll do is fast QC, and I wanted to know what this particular tool does. So in that case, what I'll do is I'll put a space hyphen hyphen help. So pretty much all this terminal based tools will be having this help option. So fast QC space hyphen hyphen help will be showing what all it can do, like this, with a detailed explanation it shows. Okay. So these are called switches. This hyphen O, hyphen V, hyphen H, hyphen cassava, all these called switches, where when you're home, at your home, when you put on a switch, a light turns on, or a fan turns on, right? Like that, it provokes an action. This switches provokes an action, or else we can call it as an argument. Okay. So it says what these arguments does. Okay. So pretty much all these terminal-based softwares will be having this section called help. If you don't know what to do, what you should do with the terminal-based software, you can just pretty much call the software. That is, if you wanted to do analysis on FastQC, just type FastQC and hyphen. If you wanted to check for its that options, simply click on hyphen hyphen HELP or hyphen H. Okay. And if you wanted to exit this terminal, simply click on EXIT. Type on exit and enter. It logs out. Okay. So these are the basic commands. Yep. So let me do one thing. If is there any video, if there is any video in YouTube, I'll just share that too, along with the data, along with the slides, or along with the things which we learned today. Okay, in the WhatsApp group. Okay, is that fine?
Okay, sir. Okay, sir.
Yeah, okay. So you're good with Ubuntu, right? Like you have Ubuntu, but you don't know what to do.
Yes,
Is
sir.
that
Fast
the
QC
case?
isn't getting installed. Yeah.
Yes,
Okay.
sir.
So I'll do one thing. I'll just if the tutorials are tougher for you guys, I'll just create a document where you can just copy paste the commands which I've given in the document, and you can paste it there at the terminal, and just you can execute it by pressing enter. Okay, if it is possible, I'll do that and I'll share the document tonight. Okay, so that it'll be easy for you guys.
Yes, sir. That will be
So
very better on initial level.
yeah, that is great. So since you guys don't know what this command does, I'll try to explain the command in the thing, or I'll just create a document where you can just copy paste the command and just execute it. Okay, so that'll be fine. And any other questions or queries, or wanted to discuss about something else?
So far, nothing, sir.
Okay, thank you, Harsh. So in that case, we'll just leave for today, and we can join back tomorrow. Okay. Uh, please refer the thing. Please refer the slide decks. If you have any questions, please ask me. We can discuss about that. Okay. And it's happy learning. And guys, take care. Good night. Okay. Bye bye.
Thank you, sir.
Okay. I'll share the recording in the WhatsApp group.
Thank you, sir. See you tomorrow.
See you tomorrow. See you tomorrow, guys. Bye bye.